feel like I need to start off with a couple of notes. Even though I kind of self-describe as myself as being a little bit more on the introverted side of the pathway, the reason I'm going to be spending a lot of this talk apparently looking at my feet is this is where the monitors are. And the second is that in no way did I just cut and paste my talk from the previous great introduction to a targeted IL-2. But you'll be seeing a lot of fairly similar data because, as it turns out, targeting IL-2 to a tumor cell can be a fairly potent strategy for getting very specific activation. So why are we interested in doing immunocytokines at all at Regeneron? Mostly, I end up talking about some of the work that my team has been done previously in CD3-targeted or co-stimulatory bispecifics. But the reason that we get into looking at these types of strategies as Regeneron is that we're very interested in developing therapeutic modalities that can potentially be used in combination with each other, especially in the oncology space. And why do we think that? That's because when you're normally looking at an immune response is not a single agent response. There are multiple signals that go into a proper cytotoxic T cell response. And I'm going to see if I can, I don't know if the pointer here is actually visible on that slide, so I'll apologize for not being able to point appropriately. But when you're looking at the natural immune cell response, say from a cytotoxic T cell, there are multiple signaling pathways that go into it. The primary, and of course the best well known, is sort of this, what's called signal 1, which is engagement of the TCR to a target that's presented in the context of a class 1 MHC molecule. This then signals to the CD3 component of the T cell receptors complex. This is really the primary signal. Without that sort of driving signal, the T cell response does very, very little at all. But it's supplemented, and it's to get the full robust response that you see, you need both co-stimulation, and that's generally referred to as signal 2. That can be through the sort of the prototypical co-stimulatory molecule CD28, but there are a number of these that are currently being studied in the clinical setting, not only CD28s, but also engagement of 4-1BB, CD2, several others as well. But then additionally, there's a third pathway, and these are just cytokines. So cytokines then provide additional support. Usually they get at least a T cell proliferation, survival, an absence of T cell energy in the presence of cytokines. And so really the natural response requires all of these signals to be present. These then can be further modulated in a negative setting through the checkpoint inhibitors, as I'm sure everyone in this room is aware. Antibodies that block or result in checkpoint inhibition or reverse checkpoint inhibition have really revolutionized the immunotherapy space. Classically, PD-1, CTLA-4, but as we've seen, there are other checkpoints that can come up that people are now addressing as well. And so it's a combination of a balance of all of these strategies to really fully activate and sort of mirror a normal T cell response. And so again, it turns out that you can mirror many of these signals through the use of biologics. So signal 1 can be mirrored through the engagement of a tumor cell-targeted anti-CD3 bispecific. Numerous approved molecules in this setting, both in the heme space initially, but now excitingly in the solid tumor setting as well. There are a number of co-stimulatory bispecifics being explored by Regeneron and others that engage these co-stimulatory pathways, either by themselves or in combination with CD bispecifics. And you can mirror signal 3 with cytokines, actually. I mean, a cytokine is a pretty good mirror for a cytokine, but it turns out that when you do that, you run into a number of toxicities, which is why these sort of targeted immunocytokine strategies are really now what people have been moving to. So what does some of these look like? I briefly had to show at least a little bit of what we're doing in terms of the bispecific space. I'm going to, I promise you I'll get onto the immunocytokine in the next slide or two. But at Regeneron, our format, there are many different bispecific formats. This is ours. It looks very much like a natural antibody. We use a common light chain format. We don't use a knob and hole substitution to get the heavy chain interaction. Instead, we make a substitution in one of the chains that allows us to selectively purify it. But the net result at the end of the day is very much the same. And we're exploring these, as I mentioned, for signal 1 and signal 2. We have a number of programs that are either now approved drugs or still in clinical development, engaging both CD3 with a number of solid and heme-based targets. But we now have this CD3 molecule, this immunocytokine for signal 3, which is this PD-1-Targeted IL-2 receptor alpha IL-2 fusion. And why did we really want to get at that? Well, one of the toxicities that you see with both the signal CD3 cytokines, signal 1 cytokines, i.e. CD3 engagement, but really with just sort of the historical use of cytokines to provide the signal 3, is that these are very potent molecules, and they have these very pleiotropic systemic effect that lead to systemic toxicity. So while they can have good anti-tumor responses, they also have these toxicities that have really limited their clinical adoption to date. And so there are a number of strategies that companies are now pursuing to try to mitigate these toxicities. And my apologies to the previous speaker. I need to add your cartoon to this slide as well. And this is in no way meant to be exhaustive, but as we've heard previously at this conference, you can have masking strategies to block the binding moieties. You can have attenuation strategies such that takes advantage of differential the differences in the tumor microenvironment, such as differential pH or different levels of ATP presence, to get engagement only in the presence or absence of those signals. As we've seen already during the course of talks today and yesterday, you can look at affinity modulation as a targeting strategy to try to get and or logic-gated approaches to really sort of broaden the tumor targeting space that you have available to you. Or you can mask and mitigate or attenuate the activity such that you get only activity in your desired setting, which is what we're doing in our context in the molecule I'm going to tell you about today, which is Regeneron 10597. This is a very, very low quality structure. So please do not try to read into this any specifics. This really is a homodimer, even though the linker lengths on one side look a little longer than the linker lengths on the other. It really is symmetric. But what we've chosen to do is to try to adapt IL-2, which is I'll talk to you about in a second. We've just had a great introduction in the previous talk. And also yesterday from Paul's presentation on targeted IL-2. So while IL-2 is a very potent cytokine to target to T cells, we've chosen a slightly different target, which is PD-1 rather than LAG-3. And we have a slightly different mitigation strategy. So rather than binding to, say, an antibody to block and then release, we're actually binding to a component of the IL-2 receptor, in fact, the component of the high affinity receptor to mitigate our activity. And so this is just a quick introduction to what the IL-2 receptor complex looks like. It's again, as I mentioned, it's a very potent stimulator of T cell and actually NK cell. Leads to proliferation, cell survival, enhancement of receptor functions. In fact, going back to the early 90s, recombinant form of IL-2 was one of the very first approved immunotherapeutics. Had activity in both metastatic melanoma and as well as RCC. But as I mentioned previously, it was associated with this severe systemic toxicities, vascular leak syndrome amongst others. And that really, again, has limited the patient population that can see benefit from this immunocytokine. So in more recent years, the field has sort of gone away from using that and looked at ways to modulate it in a way that would sort of reduce that systemic toxicity while maintaining the activity. Now, a lot of the work in the space has looked at modifying IL-2 and what are so-called non-alpha IL-2s. And these are IL-2s that are modified not to bind to the high-affinity IL-2 or alpha component of the receptor. And you can see sort of over on the right-hand portion of this slide, the IL-2 receptor is really a trimeric. It's got, on resting cells and on NK cells, you've got this beta-gamma subunit combination. That's sort of an intermediary affinity receptor system. It can signal, but what you get when you activate, say, T cells, but also on T regulatory cells, is you get activation of the IL-2R alpha subunit. This has a secret identity, well, maybe a Clark Kentish secret identity. It's also CD25, which is an activation marker for activated T cells. And what that results in is a high affinity receptor and much more potent signaling through IL-2. So again, the initial attempts in the field that focused on modulating generating these non-alpha IL-2s was an attempt to just get selectivity away from, say, the Treg populations and really wanted to expand just CD8 and NK cells over Treg cells. So why didn't we sort of look at that ourselves? The reason is sort of this experiment that we ran internally, which was that when we started to explore the IL-2 space, You used a mouse model system where we just put the IL-2, either wild-type IL-2 or one of these non-alpha IL-2s, onto an FC to just provide a little bit of half-life. And we looked at what happened during the tumor model system. So as you can see here, it's actually somewhat surprising to us that the wild-type IL-2, both either by as a single agent or in combination with PD-1, led to really good tumor control in this model system. But conversely, the non-alpha IL-2 did not. And so we really saw no tumor control with this molecule, despite the fact that when we looked sort of systemically at what's happening with the T cell populations, the molecules were kind of behaving as advertised. So systemically, when we took a look at what's going on in the spleen, you can see that the wild-type IL-2 FC really did result in a fairly profound upregulation and expansion of the Treg population. We saw less expansion relative to the non-IL-2. They were fairly similar when you looked at CD8s or NK cells. The NK cells look a little bit bigger, but statistically it wasn't significant there. Conversely, however, when we looked in the tumor, we saw something completely different, which was that we really didn't see this upregulation of the Treg cells in the tumor environment. In fact, they were a little suppressed in that setting. But we did see a very specific upregulation of the CD8 population from the wild-type IL-2, whereas the non-alpha IL-2 tended to increase the NK cell population in that setting. And so what we reasoned here is that this alpha responsibility, the ability to engage the IL-2 or alpha component subunit, was important for activating the subset of T cells that were actually tumor responsive, right? You can imagine that if you just expand any T cell, the vast majority of those aren't going to have the anti-tumor specificity that you want. But those T cells, when they do react to the tumor, upregulate the alpha component, and those are the ones we want to get at. And another thing that they upregulate is PD-1, which is, again, both an activation marker but also an exhaustion marker. And so that's why we chose also to look at PD-1 as a targeting moiety, the thought being that if we can target an attenuated IL-2 to these PD-1 expressing cells in the tumor microenvironment, that's the population we really want to engage. And so that was the basis of this design. So how we went about doing it, again, a little different than what was done in the previous talk, but the goal is really the same, which is to have a molecule that is systemically masked, and in this case we chose a component of the IL-2 receptor system, high affinity IL-2R alpha, that binds to IL-2. We tethered that, we fused that in line to the C-terminus of our anti-PD-1 antibody, and then we fused that to IL-2. And it adapts this sort of trans conformation where the molecules trans-sequester each other. But it's not a permanently locked down. This thing does open up and they do naturally release. And you get about, it says less than 1%. It's about a 0.2%, 0.1% of this species is sort of in the open conformation at any given time, which when you're looking at this systemically doesn't really result, and I'll show you some data for this, in appreciable signaling even on cells that express the high affinity receptor. But when you target it and tether it to the cell surface, you get a greatly increased local concentration such that when those cells do sort of open up, they can then engage the receptor on those cells, and you do get signaling in that setting. I said I'd show you some data. Yeah, here we go. So if you take a look, and this is, for this part, ignore the PD-1. This is all in PD-1 knockout cells. Really what we're just doing is looking at whether or not we're seeing binding to the surface of cells that express either the beta-gamma receptor or the high affinity. So if you look, beta-gamma, we're comparing to wild-type IL-2 versus this masked IL-2. You see a modicum of binding with the wild-type IL-2 to this low to intermediate receptor beta-gamma. Really don't see very much at all. The masked doesn't interact with those cells, at least as much as we can detect by flow. When you express the high affinity receptor, now you can see a greatly upregulated ability of the wild-type IL-2 to bind to the surface. And when you blow that axis up, you do see that now there is a little bit of binding of our masked construct. When you look at signaling, again, you see the same result of what you would expect. You see a very potent shift. And again, even though we didn't see signaling, we didn't see binding, it is clear by the signaling assays that we get a 3 to 4 log shift in signaling activity. So there is some amount of ability to interact even if we couldn't detect it by flow. With the high affinity receptor, we still see that same sort of signaling differential. It's a little contracted and everything is left shifted because the high affinity receptor is the high affinity receptor. But again, we see a much broadened therapeutic index in terms of what signaling we can accomplish with this masked molecule. Same thing works now when we target it, and now here's the sort of the data that shows that now when you target it, you can sort of, recapitulate the high degree of signaling that you want in the targeted setting. So in this setting, now we're looking at the differential between a knockout cell and cells that express either sort of a medium or high level of PD-1. You're looking at now either an untargeted, in both molecules in this setting, they're both masked. One is targeted, one is not. So without the targeting component on the cell, you see no difference between the targeted and not in terms of the ability to signal. But when you start to express higher levels of PD-1 on the surface, you see a progressive shift of the ability to signal out to 4 to 5 log differential in this case. We also looked at this in sort of a more normal rather than a reporter assay in normal human PBMCs by phospho-STAT5. And again, you see this differential recapitulated where you see on the targeted PD-1 expressing cells, when you sort those, you see this ability. It's down to about 200, 300-fold there, which again is sort of right in line with what we thought about in terms of the percentage of this complex that is open. But again, you start to see a greater potential for a therapeutic window using this molecule. All right, so does this preferentially expand? Yes, when we look in circulation, we see preferential expansion just of the PD-1 positive CD8 T cells over what you see with just sort of the wild type IL-2 alone. But more importantly, and again, this gets back to what was also mentioned in the previous talk, what we don't see is any real evidence of systemic toxicity in these animals. And so this is an animal where we've actually humanized the PD-1, so it's not a transgene, we just went in and humanized PD-1. So if you look with the wild type IL-2, whether targeted or not, if it's not masked, you start to see these body weight changes and also evidence of pulmonary edema when you look at the weight of the lungs in these animal model systems. We don't see any evidence of that when we use the masked forms, either in the targeted or untargeted setting in this context. So I think that we have a molecule here that we can get a very target-specific activation of T cells. Doesn't show evidence of the systemic toxicity that a wild type IL-2 would have. Does this have the activity that we want? I probably wouldn't be talking to you if it didn't. So we've looked at this in a number of different tumor model systems. Some of these we have humanized the animal model system for PD-1. Others where these syngeneic tumor model systems were not really conducive to the strain background. We've made a surrogate with a mouse PD-1. In both cases, the IL-2 alpha masking component is the same, however. But what you see here is that you see actually a good single agent activity in this MC38 model system, which is a tumor model that does have some innate immune response that we can then enhance with this PD-1 that really leads to both control of cell growth and again, a greater overall survival in these animal model system. We looked at a number of different model systems, not just the MC38 model. Again, this is where some of these, if you'll notice in the figure legend, have the murine PD-1. Again, that's a surrogate where we took a mouse anti-PD-1 and fused the same C terminus. It has a relative similar affinity to murine PD-1 as our anti-human PD-1. It's also important to note for a little bit later in the presentation that we took an epitope that would not be competitive with our own approved anti-PD-1 drug, cemiplimab, because we wanted to preserve the ability to use that in combination. And our thinking is that these molecules, even with the attenuation, may not be able to be dosed as highly as you would require for full blockade of the PD-1-L1 interaction. And so we didn't want to have to rely on this to be our blocking agent as well. But you can see here, again, across the board, both in these single agent type studies, we really do see the ability to get both tumor control and tumor remission, and you get some amount of tumor-free animals in most of these systems. Again, it varies from system to system. It's not perfectly regressing every tumor in every setting, but I think across the board we saw a really good potency of this molecule in combination. So we also took a look at what cells were doing this by depleting either CD4s or CD8s or NK cells, and the observation is this effect was really driven by the CD8 cells in these populations. We also, again, wanted to sort of further explore what's going on in the tumor microenvironment, so we did some single-cell sequencing studies, trying to take a look at what happens several days after dosing. So in this setting, we went back to the MC38. This is humanized PD-1, so it's using our clinical asset in this setting. We established tumors about 200 micromoles. We dosed twice, and after 3 days, while there were still tumors before they started to regress, we took those tumors out, pulled out cells for single-cell sequencing, and took a look at what was happening with the populations. And generally speaking, what we saw was that we saw with the treatment aspect, and this is where I wish I had my pointer, but in the lower right-hand portion of the slide is the treatment population. You can sort of look compared with the upper left-hand side, which is just treated with isotype. And you can see what happens just with PD-1 and the non-targeted control as well. But generally speaking, if you look lower right to upper left, you see a great decrease in the monocyte macrophage compartment and upregulation of the various T cell compartments, as well as a bit of the NK cell compartments in the tumor microenvironment, which is, again, to some extent in line with what we expected to do, seeing that when we looked just at CD8 expansion for this. Again, took a slightly deeper look at sort of when you sort of look a little bit more closely at what's going on in the T cell compartments. Actually, what we do again is you see a expansion of CD8 positive cells, primarily across actually a range of effector cell populations. So it's not one specific cell set per se, but a number of the different effector populations were expanded through the use of this targeted PD-1 IL-2. And importantly, you also sort of see a slight decrease in the Treg population represented in the CD4 subsets in these settings too, which again sort of aligns with what we saw earlier on when we just sort of looked at those initial studies with the IL-2 versus the non-alpha IL-2 setting. So I'm now going to sort of circle back to that very first slide that I showed as to our overall strategy as to we wanted to have these things be able to combine with other assets to, again, more fully mirror what might be naturally occurring in an immune response. And so in this setting, what we saw was that you could actually combine this with an anti-PD-1 to see an enhancement of the anti-tumor response in these animal model systems. When you look sort of early on, there's not necessarily as much of a difference. So in this setting, we combined just an isotype control with an anti-PD-1. As I mentioned, we designed these so that you could add on PD-1 without potentially impacting our ability to bind to the target in this setting. And while in the sort of initial tumor growth amounts, you don't really see a significant response in terms of the size of the tumor, when you run those studies longer and ask for the overall survival in these animal model systems, now is where you start to see actually a real differentiation by adding in PD-1. It really does look like it is a potentially combinatorially active with the targeted IL-2. In addition, we have, as I mentioned previously, a number of anti-CD3 bispecifics that are currently being studied. So we also look to see whether or not this asset could combine with our, say, for example, a MUC16 by CD3 in a solid tumor setting. And so as you can see here, we used a model system, and this is now where we've actually double humanized the mouse for CD3 MUC16. So this is, again, a syngeneic model system. In this setting, because we humanized the other components to enable the CD3, this is where we're using one of our murine surrogate molecules for this particular study. But in this study, we saw good control with the MUC16 by CD3, but we never really saw clearance in this particular model system. Now, however, when you add in additionally the PD-1 targeted IL-2, now we can drive tumor clearance in 4 out of 5 of those animals. And then finally, and this is something that actually wasn't in the paper that came out last year, so this is a little bit new data. We've also started to look at these in combination with some of our CAR Ts. And we showed, again, there's an example of that when we published this last year, but this is a little bit newer data where this is a MAGE-A4 TCR-T, which is targeted for a tumor neoantigen that's sort of well known in the space. This is a protein that's upregulated in tumors and presented in the context of MHC, and you can design TCRs or antibodies to recognize this. As you can see, when you look at what happens when you use a CAR, or in this case a TCR, I keep saying CAR T, it really is a TCR-T in this setting. Those cells, they are living drugs, they respond as T cells, so they upregulate PD-1. And so we asked the question is, could we get better response if we combine this TCR-T with our PD-1-targeted IL-2? And again, we actually saw really good results with that. So the TCR-T itself is actually quite potent. You see, initially you see a really good control and regression of tumors in all of the animals in the study. But eventually, those tumors start to relapse and you lose control in most of the animals in this setting. When you combine this TCR-T with a control that's just non-targeted IL-2, you don't really see any added benefit. But now when you target it, combine it with our PD-1, IL-2, IL-2R alpha, we saw tumor control in all of the 8 animals in that study, and it maintained it out to the end of the study. We didn't see any evidence of relapse in that setting. So I've got just a couple of minutes left to quickly go through the summary here. Overall, what we've showed you is, again, we've generated this Regeneron 10597. It's a PD-1-targeted masked IL-2. The goal is to deliver specifically an IL-2 to T cells in the immune microenvironment that have been activated and are hopefully our anti-tumor T cells. It does have attenuated activity, so we don't see any of the systemic toxicities that you would with a wild type, but we've maintained that IL-2R alpha, that binding ability that we think is important for the ability to get the full response that we want. It can have single agent activity, but again, more importantly, we've shown that it can be combined with a number of different therapeutic modalities. And based on all of that, this entered phase 1 clinical trials last year. We're currently still in sort of dose escalation stage, so I don't have any clinical data to share with you at this time. Hopefully, at some point in time in the future, I'll be able to share some of that, but that's where we are right now. And so at the end, I'd like to thank all of the team at Regeneron who put all of this together, because it certainly wasn't me. Specifically, Tong Zhang and Chris Wu and my team who really led this effort in engineering and developing this molecule along with the rest of their group, supported by the rest of the bispecific. And we work very closely with all of our colleagues in the OncIO and therapeutic protein teams, as well as the I&I group as well. And also, I'd like to thank the various folks in our molecular profiling team who did all of that great single-cell sequencing work that I was able to tell you about. With that, I think I've got about a minute left for questions, and thank you for your attention.
Andrew, please go ahead. Yeah.
So it might be a difficult question to answer, but do you have any idea what the active concentration of the IL-2 in the tumor is, how that compares to serum IL-2, plasma IL-2?
So we haven't been able to measure yet, or at least we haven't. I don't have good numbers for you for what the active concentration is going to look like, certainly in the clinical setting. So unfortunately, I'm just going to have to sort of pass on that because I just don't have the data.
Have you compared your masked IL-2 with the IL-2 mutant, which was designed to specifically activate like CD8 T cells?
Can you repeat
IL-2
that?
mutant. There's a engineered IL-2 form which was designed to specifically activate CD8 T cells, but not the Treg and NK cells. Have you compared to your masked IL-2 with the IL-2 mutant?
The only comparison that we've done is the one that I showed you with the sort of very beginning, where we used a comparator molecule from the literature that had this attenuated non-alpha IL-2 binding phenotype. We've looked at doing a targeted form of that as well. We used, again, that same sort of molecule for the literature, but we didn't mask it. We just used it as a targeted form to PD-1. And we saw our molecule worked, slightly better than that and had slightly better safety profile. We don't necessarily fully understand what that differential is, but we're looking into that a little bit more closely. Is that close enough to what you were asking? If not, you can find me afterwards.
It's pretty similar to the one, just basically the other mutant tried to avoid the binding to the IL-2 receptor alpha,
Yeah. So
Okay, so one final question.
Okay.
Go on, please.
Reason to use IL-2 over IL-15 in the context of your alpha chain results, or have you kind of thought of both of them?
We've thought about, honestly, we're looking at other cytokines as well. We started with IL-2 because it did have sort of clinical validation from those earlier approvals, and certainly is was known to be very potent. But we also, I know other people are exploring IL-15. It's have fairly similar, and especially as you mentioned, with our strategy, there's mix at like the IL-15 sumo domains, and there are similarities to what's going on in the IL-15 space as well that we're also looking at. And there are other cytokines that you can envision this strategy might also be able to be worked for. So, but no, mostly we had to start somewhere, and this was the one that had, again, good previous clinical validation, so that's where we started, and again, it worked quite well, and so we went with it.